Well, everyone, I hope you're here now because we're going to begin this talk as part of ScienceFest 2020. I just want to say there are many things on the ScienceFest schedule this year that are all virtual, so I hope you'll find them and join in with us. The full schedule information and more details about ScienceFest 2020 can be found at losalamosciencefest.com. This event will also be recorded and be found on the website afterwards. During the talk, please go ahead and use the Q&A button on Zoom to ask questions as they come to mind for you. We will take questions at the end of the talk, but we want to get your questions to us whenever they come to mind for you. So please do that. All right, so again, welcome to this talk, The Shape of Your Genes, Using Supercomputers and Strawberries to Understand How DNA Works. This talk features two researchers, one of them a good friend of mine, and I will introduce her first. Oh, let me say thank you for joining. I'm Linda Deck. I'm the director of the Bradbury Science Museum here in Los Alamos. It's the Museum of Los Alamos National Laboratory, and one of our speakers is a researcher at Los Alamos National Laboratory and has a wonderful exhibit in the museum. So I hope you can come see that someday when the museum returns to being open. So this researcher, who I am going to introduce now, is Karissa Sanbonmatsu. Karissa Sanbonmatsu is a structural biologist at Los Alamos National Laboratory, where she leads the Sanbonmatsu Laboratory established in 2001. Her team uses computational and experimental approaches to understand the mechanism of a diverse array of epigenetic and non-coding RNA systems, including chromatin, ribosomes, ribose switches, and long non-coding RNAs. She has been a leading figure in structural studies of long non-coding RNAs in epigenetics and is currently studying the mechanism of epigenetic effects involving chromatin architecture. Her body of work led her to be elected as a fellow of the American Physical Society in 2012. Most recently, her group set the record for the world's largest published biomolecular simulation at 1 billion atoms, the first simulation of an entire gene. Her TED Talk, The Biology of Gender, From DNA to the Brain, has over 2 million views on TED.com. You might like to check that out in your free time. Now let me introduce her co-presenter, Dr. Anna Lappala. Anna completed her PhD in physics at the Cavendish Laboratory, University of Cambridge, in 2015. Shortly thereafter, she worked in the Department of Theoretical Chemistry at Cambridge with Professor Don Frankel, FRS, as a Newton Trust Fellow, after which she moved to New Mexico and is working at Los Alamos National Laboratory, or worked at Los Alamos National Laboratory as a postdoctoral Center for Nonlinear Science Research Fellow. She's currently an instructor at Harvard University, where she works on soft condensed matter theory problems. So without further ado, I'm going to turn it over to Dr. Sanbonmatsu and Dr. Lappala. Karissa and Anna, welcome, and we're looking forward to your talk.
Well, thank you for that introduction, Linda. It's great to be here together with Dr. Anna Lappala for ScienceFest. We're really excited that we can keep this going over Zoom. And so today we're going to talk about DNA. So we know that DNA, it's inside your body. It's a long string-like molecule. They're many copies of this molecule in all of your cells. And inside that molecule are the instructions that make up your entire body. So it tells when fertilization begins, before you're even born, it tells that fertilized egg how to transform into an embryo, into a newborn baby, and then it allows you to keep living and do all the things you need to do throughout your entire life. And even though we know the entire sequence of DNA, we don't really understand exactly how it works. And so I'll show you a few slides here. Okay, so in this movie animation, we're showing a small segment of a chromosome here in pink. And what we're trying to do is understand how these chromosomes work. So there's a fundamental question in science right now, and that is that if every cell in your body has identical DNA, how can the cells be different from each other? They all have the same DNA sequence. How can they be different? So for example, once in terms of the embryo development inside the womb and becoming a baby, the way it works is that the fertilized egg divides into 2 cells, those divide into 4, and so on until somehow you get a newborn baby. And the baby will have different kinds of cells like muscle cells, brain cells, neuron cells, intestinal cells, and so on. These are very different kinds of cells. The neurons in your brain look completely different than the muscle cells, and they do completely different things, but they have the identical DNA sequence. So how is it possible that the DNA is supposed to have the instructions for everything, but it's identical in the same cells, yet the cells come out completely different? Well, the answer is that the genes are turned on and off differently as the embryo develops. And we're trying to understand how this works.
So Karissa, you call this epigenetic programming. What is that? And are the genes the software engineers of the cell?
Well, that's a really good point. And so epigenetic programming, it's sort of, it is sort of like software. So the idea is that they're essentially identical genes in every cell, but those genes are turned on and off in a different manner. So at different times as the embryo is developing, different genes are turned on and different genes are turned off. And this, you can think of this as sort of a large circuit or almost a computer program, and this we call epigenetic programming. So yeah, you can think of it as software engineering. So the way this works is that you have this DNA inside each cell. It's a really long molecule. In fact, there's enough DNA in your body to wrap around the entire Earth many times. And so what happens is all that DNA is compacted somehow into each cell, but it's done so in a very organized way. And the way nature evolved to organize that compaction is by producing thousands of these little tiny spools. Now these are nanoscopic, so you can't see them with the naked eye, but somehow the DNA, it's wound around each of those spools. And the spools kind of are always undulating and winding and unwinding, as the cell is moving around, and they somehow control how genes are turned on. This is what we call gene expression. And what people believe is that when all the spools are wound up tightly and compacted, the genes are shut off because the machines needed to use those genes can't get at the innards of that compact blob. However, when the genes are spread out, then they're more easily accessible and they can be turned on. So I'll show a brief kind of mock-up model here that I have manufactured out of a garden hose and a garden hose spool. So I have this set up here. So I just got to turn off my timer. Okay, so here is this piece of DNA. And what we have here, sorry, I'm still having trouble with the timer. Okay, okay, there we go. So we're visualizing the DNA by this garden hose spool. the garden hose wrapped around the garden hose spool. And there are 4 bases in DNA, adenine, guanine, cytosine, and thymine. And I've represented them by taping different Post-it notes along the hose. And you can imagine that if this were scaled up, we'd need enough hose to fill up probably the entire town of Santa Fe to represent what's in the cell. And so this is the DNA. And then a segment of DNA is called a gene. So basically you have a long segment of the DNA, and that's called the gene. And then the molecular spool is here. And this is called the histone. So the histone is the molecular spool. And what happens is that when the DNA is spread out, in that case, then the gene is turned on. So the machinery can grab onto this DNA, read it, and then make proteins of what it needs to do. However, when signals trigger the gene to shut off, what happens is the DNA wraps around these spools like this. And so in that case, the machinery needed to turn on that gene can no longer get at the gene. And so in this case, the histone is turned off. So that was my brief mock-up of this story of how DNA is turned on and off. And so now we'll go to the next slide. And so in this case, what I wanted to say that this process of compacting the DNA and expanding the DNA is really important for different kinds of diseases. So it turns out that there are special molecules that help control the turning and on and off of these genes. And these are very important for different kinds of medical conditions. So Rett syndrome is an important drug target for intellectual disability. There's another molecule complex called SWI/SNF that controls how the DNA slides along the spools, and sometimes the spools can even get ejected. And this is a cancer target right now.
So Karissa, what about the coronavirus?
So that's a great question. The coronavirus is something affecting all of us now. It's in the news every day. And there are some important points about coronavirus. And it turns out that the way the immune system responds to coronavirus is different for everyone. And they're finding that different blood types are more resistant to coronavirus than others. In addition, there are certain genes that are affected by coronavirus. And so we're hoping that if we can understand how the genes are shaped and how they compact, we might be able to understand why some people are affected and some people are not affected. Like we know that older people are affected more than younger people and so forth, but no one really understands exactly how this works. And that's one of the things we're interested in now. So to look at this in more detail, what we're doing is we're simulating regions of chromosomes on the supercomputers at Los Alamos. And so here we're showing one of the main supercomputers. And what these are sort of, they're essentially 1 million computers or 1 million processor cores hooked together in a giant warehouse with a high-speed fiber optic network. And this enables researchers like us to do big calculations. And so what we do is we do something called molecular dynamics simulations. And we're essentially solving the laws of basic physics or Newton's equations for every atom in this biological system. And we're trying to understand the forces on each atom due to all the other atoms. And that way we can get a small glimpse of what happens in the life of just a small piece of DNA. So if we wanted to simulate atom for atom what all these pieces of DNA are doing inside an entire cell, we need around 100 trillion particles or a quadrillion iterations in the supercomputing code. And for this, even the largest computer on Earth is not nearly enough to do this. So as a start, we're taking the first step of just simulating one single gene, atom for atom, and seeing if we can learn anything. And so here we're showing one of the calculations we did on the Trinity supercomputer at Los Alamos National Laboratory. This was a collaboration with Anna and with a Japanese group in Tokyo, Yuji Sugita's lab, and a group at NYU, and many people around Los Alamos National Laboratory. And we're showing one single gene. And here, if you look carefully, you can see this double-stranded blue DNA, its helical nature, and it's wrapped around these spools, these gray blobs. And those themselves are made of proteins that themselves have little helices in them. And so we're trying to look at the nanoscopic environment in these crowded regions of chromosomes to see what the electric fields are like, to see water crashing into the DNA, and to see how this can compact and expand and so forth.
So Karissa, how long did it take you to simulate those 1 billion atoms?
So this was a large project. It took from beginning to end about 5 years, and the actual computer runtime, it took many months on around 500,000 cores in the Los Alamos supercomputing laboratory. Okay, so that's what I had to say about these supercomputing simulations. So now I'll turn it over to Anna, who's going to tell you some more.
Hello, my name is Anna. Thank you so much, Karissa, for your beautiful talk. So first, I wanted to start off with a brief experiment. So to feel more like a scientist, I will wear a lab coat. And so what we have here today is, instead of strawberries, which we promised, we have tomatoes. And so we will be planning to extract DNA. We'll be trying to extract DNA from tomatoes. And here in a Ziploc bag, you see a sludge, this liquid with tomatoes in it. About 2 tomatoes went in here. And also what we used as a solution to break down the cell walls and the lipid bilayer, we used soap with water. In order to aggregate proteins, to get rid of proteins, we use salt. And so here, this solution contains all of these ingredients. So it is something that you can easily do at home. And so what we will do now is just pour it into a little jug here. So hopefully this works. Okay, so I'm just pouring this into a jug, which will be easier to handle. Okay. And now we need tissues and a glass. So here I have a very clear glass and some paper towels or paper tissues. So I will just use 2 just in case they break. And I'll make a little dent like this. So, but no holes, make sure there are no holes. And so the tomato sludge is actually going to go inside. So we're just filtering out all the stuff that we have from the tomato, and we just want the liquid. That is where our DNA is going to be. So hopefully this works. Okay, so this is what I'm doing. I'm just trying to get that juice through. And I'll need a little bit more paper towels, so just bear with me a second. So another paper towel, and more of the sludge is going in. So this will filter out all the bulk that we're not interested in, because all we want is a solution. Okay, and I will show you what's happening right now is that it's filtering out, it's being filtered, as you can see. And so it will be a rosy kind of colored, clear solution. So I will stop for a second, because what we need is to let this go through. So as it's being filtered out, you can see that everything is working. Okay, so I think we won't need much more of this right now. Okay, so we can leave it here. So here's our clear solution. There's nothing inside. It's kind of just rosy colored, right? So the next step is to precipitate our DNA. And for that, we're using alcohol. So this is cooled alcohol. I kept it in the freezer for a little bit. And so what this will do is precipitate. So this is, I can show you, this is just 70% ethyl alcohol. And so what I will do is just pour this alcohol slowly into our tomato solution. Okay, so over time, what you will see is that we will form a gradient within the solution, and all the heavy stuff will be on the bottom of the glass, and all the less heavy stuff will actually be on top. So your alcohol will be found somewhere here, and that is where the DNA will also be. But we need to wait for that to precipitate. So let me give you a talk just to briefly talk about the structure of DNA. So I will be sharing my screen. Okay, so can we model a whole chromosome? This is a question that is something that Karissa and I and Jeannie Lee here at Harvard, we have been working on for the past year or so. And so I will be sharing with you how this can be done. So in order to model a DNA chain, for example, what you need to do is build a model, a computational model, where each particle, as you can see on the right-hand side, the upper figure, you see those blue little particles. So these particles represent, say, a base pair, and this is just a single unit of your DNA. So for example, here you have 1,000 of these spheres in a beautiful spool in a structure that's spherical. And in fact, in many viruses, this is how the DNA or the RNA even is packed in this particular structure. So then what we also have to consider in our model for the beads to be connected, because we want to form a chain, a string of these little particles that are called, in polymer physics, these are called monomers. So each particle has to be connected, and this white segment here is showing you the spring. And so it literally acts as a spring between two particles, and it fluctuates whenever you're deforming the chain. So all particles interact with each other. So even if they're not interacting by a spring, they interact through a different potential that allows the chain to either expand or form a very, very tight knotted structure, so to collapse or fold, as we call it. The flexibility of the chain can also be controlled. So that requires for us to have an angle. So if you have 3 different particles that you are interested in, between those 3 particles, you will build an angle. And this angle can be either very flexible or very rigid. So you can, this can be set in your simulation such that you replicate the experimental evidence for how stiff the DNA is.
So Anna, when you say you're making a model, what do you mean by that?
This is a very interesting question, Karissa. So when we build models, we want an approximation of reality. We want to know how something behaves without going into detail. The models that Karissa is building are on an atom level resolution, and this is absolutely amazing that we can achieve this. However, if we were to think about building a whole genome, this would be impossible, just like Karissa said, to do on an atomistic level, atom by atom, trying to model your chromosomes. This, at least with the computational abilities that we have now, this is still impossible. So we have to make approximations to make a simpler case for something that is extremely complex. So for example, here we're looking at an electron micrograph of a bacterial chromosome, and this is E. coli, a very common bacterium. So this is just a literal picture of what bacterial DNA looks like if it's all opened up. And as you can see, it's very random. It's not built in this beautiful spool, and it's not very clear that you could just use the spool as a model for this particular molecule, for example. So in order to...
So Anna,
oh, yes?
Another question. So are these bacterial chromosomes different from human chromosomes?
Absolutely. So most bacterial chromosomes are in fact circular as opposed to linear, like our chromosomes. And usually we only have one chromosome in a bacterium. So this is very different from, say, humans or mammals or higher organisms. So if we want to design something that resembles the bacterial DNA, we actually want to study something that's the initial structure of which is very random. So we're able to build these random structures using the self-avoiding random walk algorithm. And what it does, it simply builds a chain such that the chain does not cross itself. So whenever you build a structure particle by particle, and you make sure that it doesn't cross itself, and that way you get an open, beautiful kind of coil in your chain. So this is what happens if you add salt to the solution. It starts folding. So again, I will show you the movie where first you form these little blobs, and these are almost like rain droplets, right? You have these little droplets then that coalesce, fuse together, and then eventually form the lowest energy structure that's obviously a sphere.
So is that structure completely random, or is there are there any features of that?
Absolutely. So since we, in this particular model, looked at a generic chain, which could be random as you want it, we're actually, as a result, getting a very randomly packed chain where all the beads are able to interact with all the other beads. So all the segments of the chain are able to entangle and interact with each other. Now, of course, in the chromosome, this is not the case. Because if you think about the chromosome, it needs to function. It needs to have certain genes turned on or off. depending on where the chromosome is located, for example, which cell type it's in. So because of this, everything in our genome and even in bacteria is very, very highly regulated and structured. So even though this structure looks like a bowl of spaghetti, or maybe just one spaghetti kind of piece folding on itself, our genome is not like that. So here is an experimentally derived structure of an entire chromosome of a mouse. And so here you see I've colored this chromosome differently. One part of it is pink and the other one is white. So this is just to show that the chromosome ends do not actually mix in the real chromosome. And this suggests that the structure, even though it does look just like a spaghetti ball, you won't be able with a naked eye to tell whether there's a difference between a randomly or organized chain or a non-random chain. But here, just looking at the ends, which do not mix between themselves, you can see that the structure is not random. Of course, there are many more ways of identifying that this is the case, but this would be too much detail for this talk. So,
so what is the pink part and what is the white part on this?
Right, so the pink part is just the beginning of the chromosome, and the white part is the end of the chromosome. And here, as you can see, that there's no mixing between the two. And so the way everything folds is such that you fold one end in kind of one blob and the other one end into a different blob. But these blobs are still very closely packed together. So they're very tightly folded. Of course, this is not the only thing that you can do. You can also 3D print a whole chromosome. And this is a picture of the chromosome, but here I can actually show you the real thing in my hands right now. This is the same chromosome that we just looked at. And unfortunately, a gene just broke off. This would be the size in relation to this whole chromosome. This gene, the piece that just broke off, would be relative to what Karissa was talking about, the GATA4 gene that she simulated with 1 billion atoms. So the scale is very clear that a single gene is a very tiny segment of this whole chain. So this is the end of my presentation now, but I will show you the results. And hopefully you can see that on top, we've formed this very strange kind of snot-like stuff. It's very slimy. And this, in fact, is the alcohol part, so it starts somewhere here. And the DNA clearly separated out on the top because of its density. It's not as dense as everything else that was in that solution that went down. And so now we have this snot on top, which is effectively the DNA of your tomatoes. So sacrificial tomatoes actually did a great job helping me with this experiment. Thank you very much. So that's the end of my talk. Thank you.
Well, Anna and Karissa, that was fascinating, and love the analogies and the ability to help us understand scale and complexity. And there are loads of questions about this. So I'm going to actually throw out one that came to mind for me, and I think it was something that you said, Karissa, which was something about, oh, you were talking about coronavirus and and suggesting that it's something good to study the expression of the genes of people in responding to the coronavirus and how we differ in our response. And you mentioned that age seems to play a role in how we respond to the coronavirus. I'm wondering, do we know much about gene expression with respect to age? Has there been much research about how that works?
That's a great question. And there is research on how aging affects gene expression. And there are many, many effects like the ones we've been talking about how the sort of shape of the genome affects gene expression, or we call that epigenetics or epigenetic programming. And then as far as the relation to coronavirus, coronavirus is so new that people are really just trying to figure out how it works right now. Most people are focused on the virus itself, and very few studies on the actual effect on genes or the gene response have been done. I think there's only one published study to date so far. So it's really an exciting open area for research.
So no one has really done research on any of the SARS type viruses?
There have been previous studies, and they do see effects on the other SARS type viruses, but not much on the corona SARS virus.
Thank you. I'm going to look at the Q&A and see what kind of questions. Here's another one, very interesting. Angie asked, do you know which blood types are more affected by coronavirus?
Yeah, this is the O blood type. These tend to be more resistant to coronavirus, but these are still ongoing investigations. So I believe that study was not peer reviewed yet, but they do present data for this.
How much, and I'm just going to ask the question then, do we know how much variability there is in our genes that account for our difference in blood type? Do we know much about how our blood type?
There are specific genes for blood type that have been mapped.
Interesting. All right, let's see. Anna, this one's for you. And actually, I'm going to ask a question before I ask this one because it was asked earlier. So you originally thought you'd do strawberries and you didn't. You did tomatoes, as you said. Why those two fruits? Because we actually know that a tomato is a berry, right? It is a berry. So why would you have chosen tomato or strawberry?
Yes, so for strawberries, they simply have, and tomatoes as well, and a lot of vegetables and fruit actually have more DNA in them than anyone else. So they have many, many chromosomes, and I don't want to lie exactly how many each organism has or each plant has, but they have more chromosomes than other organisms. That will just help you extract more DNA. However, you can also do the same with kiwi fruit, for example, or even with your own saliva. So there's always, the results will be different, and you probably won't see as much DNA as you can see here from a tomato if you did the same test with your saliva, but this is just a better kind of, you get more material out of it.
And your goal for your experiment that you showed us was, it seems to me, simply to be able to show us and here's the DNA all collected
That's
in
right.
this layer. So someone also asked, if you were going to extract DNA for working on for research like human DNA, how would your extraction process differ from what you just did?
Right. So, of course, for just showing DNA like this, I would probably just spit in a little tube, and then, do the same things I did. However, of course, in the lab, there are protocols to extract DNA, and they are much more. Here I just kind of had a glass of water, a little bit of dish soap, and a little bit of salt. This is, of course, not how you would do it in the lab, but there are protocols to do this so that you carefully measure everything and make sure that your proportions, everything is in proportion, and you extract it with there are kits that you can buy to extract bacterial DNA, for example, and so you won't even have to worry about what's in the kit. You know that you can just order that and extract your DNA from there. And then also, if you want to say look at DNA, you would want to spin it down in a centrifuge. One time I worked in a lab, and we used very big centrifuges with liters of bacterial DNA, and unfortunately one of the tubes in the centrifuge exploded because it was broken, and no one knew that this was the case. So because the centrifuge became unbalanced as a result, the centrifuge stopped working and started jumping around the lab. So this can happen. Science is very exciting. Just to look at your DNA, even your own DNA, you can just do a very similar example, but I do not recommend drinking this.
Sounds good. All right. So a question that Joanna O'Neill asked is the DNA collected from your experiment denatured by the process?
The DNA is not necessarily denatured by the process. What we simply want to do with the soap, the dish soap, is to open up the cells, to open up the cell walls, so the lipids that we want to kind of get rid of with the dish soap. And then we kind of do denature DNA in the alcohol a little bit, but it's not really going to be anything that we're interested in. So for example, if you would want everything intact, you of course wouldn't want to use this particular experiment. Yeah.
Right, right. We've got a good question from our good friend Donovan Porterfield, who is a colleague of ours at the laboratory. He's a chemist. He wonders, to better model genes, do we need bigger parallel computing platforms, more efficient modeling algorithms, or maybe new computing platforms such as quantum computers? And this is a question for both of you, I'm sure.
Right. So I don't know if you, Karissa, want to go first on this, since you've done quantum computing as well. I would only say that I think we could do better in all areas. We could improve the supercomputing power, but at the same time, we could also improve our algorithms, so we could develop novel algorithms to study the folding of DNA and full genomes. I think it really is a matter of time until we get there, and I think we're on the right path.
Yeah, I totally agree with Anna's answer. And so, yeah, so I think we need everything. So it's kind of a high bar to simulate an entire chromosome atom for atom. So for that, we need maybe 10,000 supercomputers of what we have today instead of Trinity. But That said, I think in the past, people have already also made statements like this, but then people working on really nice, efficient algorithms that no one could foresee, those algorithms came out, and then it really could speed up things by 10-fold, 100-fold, 1,000-fold sometimes. So I think all those things can happen, but for sure we need bigger and faster, more efficient computers. And for quantum computing, we're just in the baby steps of this. It's so early. So we hope someday we would have a quantum computer that can do biological problems in a realistic fashion, but we're just taking baby steps right now.
Right, there's a long way to go to productive quantum computing, but to be able to solve all the states at the same time, which is what quantum computing would do, would seem to be really sort of that gate opening technology to allow us to learn that.
Yeah, absolutely.
Yeah. All right, let's see, what else do I have? Does embryonic gene expression differ for cell development for different individuals, even though they would be part of the same species?
Yes, the embryonic development is very different for every individual. And so there are big differences. And then even for identical twins, you can see big differences happening. And these differences occur due to these programming effects that we've been talking about. And there's a famous experiment on these so-called agouti mice. And these were mice that, this was actually after birth, but they were fed, they're identical twins, but fed different kinds of nutrients. And one turned out to have a totally different color fur than the other one. And then one was twice as fat as the other one. So even though they're identical twins, and they were able to sequence the genes and look at the epigenetic programming marks and see significant differences on the appropriate genes. So that's kind of a canonical example in the field of how you can have identical sequence, but vastly different outcome.
So the nature versus nurture question is answered.
Yeah, so I think the answer to nature versus nurture, it's neither nature or nurture. It's a kind of fusion of both.
Yeah, fascinating. Let's see what else I have here. What happens? So, Anna, you showed us your beautiful 3D printed model with the little broken part, unfortunately. Well, what happens if a gene is broken from a chromosome in real life? Do we know?
You probably don't want that to happen in real life. I think it really depends, of course, on what gene exactly you were talking about. There's a lot of DNA that's not necessarily coding for genes. There are kind of intergenic regions in your DNA, so it's not exactly how it wouldn't be as important to miss one part. But again, if you're missing an important gene, you probably won't even be born because of that missing gene. So it really depends on which chromosome, where, and also you have to remember that you have 2 chromosomes. So sometimes there's compensation if you have gene missing on, so something happened on one chromosome, then the product of the other chromosome can compensate for that. So it's really much more complicated than this, of course, but in general, you don't want to miss a gene or an entire chromosome or have more chromosomes than usual. The kind of the ideal is what we already have.
Yes, yes. And the genes encode for everything that it takes to make us live. I mean, that's
right.
what they're for. They're the instruction set. And so if there's a missing instruction, it was probably there for some reason.
Exactly.
Let's see what else we have here. Does the virus DNA or RNA in a coronavirus duplicate the same way ours does?
Okay, this is an interesting and very good question. And so the short answer is no. And what viruses are kind of, they call them almost like parasites. And so what they do is they invade your own cells and they hijack the machinery used to duplicate your DNA, and then they make it duplicate its own DNA. And so they take over the cell, they make lots of copies of their own genome. In the case of corona, it's an RNA genome, so they make their own RNA genome and the proteins enough to assemble the whole thing. And many viruses make so many copies inside your own cells, it explodes your cell and destroys the cell. So yeah, they do. I mean, there are a lot of similarities in the replication process, but there are also important differences.
Yeah, I mean, ideally, any kind of organism doesn't want to make too much of anything that would destroy its host, or that it doesn't have anywhere to live. Is that true?
Yeah, absolutely. And this is also an interesting question because anything fundamental about the virus, like how it replicates itself, these are high priority drug targets right now. So if we can stop how it replicates itself, we can help to treat the virus.
Yes. So the more we learn about this process, the better we will be at approaching different solutions.
Absolutely.
Yes, yes. And let's see, we have one more question, and that's just about perfect. So let's see. This is interesting. If you knew you were more susceptible, is there a way to change your genetics if you happen to be someone more predisposed to getting a worse case of COVID-19? She said, I'm guessing not. And our first step is understanding why some people are more susceptible to getting severely sick.
Yeah, that's absolutely true. And the first step is to understand the genetics. And one of the reasons to understand which genes do what in responding to a corona infection is that if we can understand, say, one of the key, well, for example, if we can find someone who has a genetic mutation that's resistant to coronavirus, we can try to mimic that with a drug that we can come up with. So to have, if we can give someone a drug that has the same effect as a resistant mutation, that would be a great step forward. And but the first step is to try to understand the basic genetics of the immune response.
Very good. I have to go to at least one more question that had come in via email, because this one gets us thinking even more broadly. Could epigenetics support or explain the Jungian notion of a collective unconscious? Either of you want to approach that one?
That is a very interesting philosophical question, and I think I've been thinking about this a lot, and I think that there are certain situations where, for example, a child can start counting without them being taught to do that specifically, or can start reading without being taught. This is very rare, and it can be seen as kind of just a fluke, and of course there's not enough statistics to kind of understand that, but I would not rule it out. On some level, I think there could be something that regulates. Of course, I mean, genes regulate our brain function as well, so that probably would affect somehow the collective mind.
And so the last question that I'll bring up, because it's just so fascinating and it's along this same line. What might be the implications of an increased understanding of epigenetics in the prevention and treatment of generational trauma?
Yeah,
Could be.
there's already a lot of work in this area right now on this. And so I'd say about 10, 15 years ago, there are many studies on animal studies looking at the effects of stress on your DNA. and they found massive effects on how you respond to trauma and stress as a child, basically. But this is for rodents, rats, and mice. So this would be affect their stress response throughout their lifetime, and then actually be passed down to future generations, because a lot of these DNA marks can actually get passed down to your kids and grandkids. And then, so that was a while ago, a study from the Montreal group. And then since then, a lot of people have been trying to see how can we undo these effects somehow. And there are a lot of promising studies to look at the effect of, say, exercise, diet, different therapeutic treatments on can we somehow wipe out those marks on the DNA. So it's an important question, and a lot of people are working on this now.
I also think that malnutrition has been looked at in humans. So, for example, if during the war, the mother was malnourished, the offspring for the next several next generations actually was smaller than on average. So I think that there are certainly some hypotheses which would prove that, you know, that might affect generations in the future as well.
That's fascinating. This whole session has been so fascinating, and just like science does, the more we ask questions and get answers, the more questions that we have to look to for the scientific method and help us understand. I want to thank both of you, Anna and Karissa. This was a fabulous session, and thank you all for being part of the audience. I hope you will enjoy many more Science Fest talks this Science Fest 2020. Remember, Los Alamos ScienceFest.com to find the schedule. Thank you very, very much.
Thank you.
Bye-bye, everyone.